Wonderful. So as you know, today's session is on digital pathology. And essentially what we want to do is able to talk a little bit about the big problem, and then our speakers, Dr. Faisal Mahmood and Dr. Lee Cooper, will be giving you a much nicer overview of the specific problems. I just thought I'll try to motivate what we have going on for the audience's understanding. So when we talk about AI for integrating imaging and genomic data, pathology has a very important part to play, especially because it presents the visual side of what we might want to understand about data. With pathology, with the genomics providing the complementary genomic side. So essentially you want to combine aspects of visual pathology with aspects of molecular pathology, both of which can be either correlated, which is what this double-edged arrow is supposed to mean, or supposed to integrate the visual pathology with the molecular aspects. And so the questions that might happen in the interface of these things are extremely interesting. Now, the whole premise of this idea of AI tools for integrating come from the notion for the need for reliable decision support for digital pathology. And we know that pathology slides like this, you see an H&E image of tissue is typically used routinely in a clinical setting. And people use these kinds of things for disease grading, for immune activity assessment, for metastatic cell assessment, etc. And essentially, the number of volume of these kinds of slides that are acquired in the clinical enterprise end up being the most useful for sort of motivating the need for AI engines in this regard. And there are clearly some very nice correlates for that kind of need, because not only can you help a medical center do a more reliable and robust job processing its data, but also if these tools become truly vetted, the community might be able to adopt them for a much more sort of a global sort of perspective on how to leverage AI solutions, even in a low, medium income kind of resource settings. So there couldn't be a better time to do AI for pathology, especially given the huge amount of data sets, because you've got a variety of databases like TCGA, CPTAC, the Cancer Imaging Archive, all that have been enabled. So it's very big global projects to be able to motivate the need for AI. So one aspect where AI might be able to make sense of all of this is in the case of routine H&E images. The other could be in the case of multiplexed data. So the idea is to be able to use AI technologies for knowledge discovery and for making things more scalable and standardized. So I'll give a very few case studies of AI in pathology very, very broadly at 30,000 foot view. And then our speakers will give you a much more nicer version of these specific case studies. So one aspect where one could use AI is for things like content-based image retrieval for image mining assessment from these very complex slides. So you could get a satellite view of these different slides. You could do navigation. You could essentially use this like a map, if you will, and try to figure out what's going on in the satellite view. And not only can you use this in a way to discover knowledge, but also use that as a way to say you want to look at a particular area and find other areas like it, etc. So it helps in the automated grading of slides in similar ways as well. The other aspect might be, as I said, one was the content-based retrieval. This one's the notion of trying to integrate and methods to mine the combination of visual pathology with molecular pathology and build more reliable and perhaps prognostic integrated engines that can leverage the best of both worlds to build better disease grading algorithms or better prognostication algorithms. Now that you have the ability to, the third study could be, now that you have genomics and you have visual pathology, one could try to correlate these two and ask, well, what molecular pathways underlie these complex morphological behaviors? So you could look for network. This is an example of a gene network that underlies these complex topographies and morphologies. So that is the power of being able to integrate, again, imaging with molecular data. One more aspect that has become very, very popular in the past couple of years is using image processing algorithms, both regular machine learning or even deep learning algorithms, to be able to virtually stain H&E slides like this with different markers of interest. You could stain for a mutation, you could stain for immune status markers, Or in the case of colorectal cancer, you could stain for microsatellite instability or MSI. And again, this gives you a very powerful paradigm to be able to visualize complexities in the tissue by virtually staining these H&E slides with different markers, and then essentially aid the process of molecular assessment. As you can imagine, this is a beautiful area for the integration of these different data sets and for the examination of multimodal data fusion algorithms. There are aspects from machine learning, like structured data regression, that could be used in these kinds of problems because there is correlation, as you might imagine, between the outputs and the inputs. And so people use ideas like multitask learning or sparse structured regression to be able to make these mappings between imaging and molecular data. As you can imagine, computer vision algorithms for image processing are a very basic yet very high research sort of need. And people are also trying to figure out ways to bring the human expert into the loop of learning. So essentially, you have humans participating along with the AI algorithm to be able to do interactive learning from these kinds of data sets. And so there's a lot of room for interactive learning, machine learning loops. So as you can imagine, this kind of an area is a perfect opportunity for researchers from machine learning, from analytics, systems biology, statistics, to be able to come together and sort of think about how one might bring their toolkits to solve this kind of meta problem. In the process of trying to do this, some key considerations might emerge. For example, what do these features mean? What are we, when we use deep learning and really deep networks in terms of architectures, what are we learning, what are we measuring, and how can they be interpreted? These are natural questions that arise. The second question that arises is, once you build an AI engine and you get certain metrics of performance, how does one meaningfully adopt them in a clinical workflow, right? There are uncertainties prevalent in the data, uncertainties in the model, uncertainties in the way these inference algorithms work. How does one change these uncertainties to build a better clinical workflow? And then how can we use this kind of information to derive knowledge that can complement human insight, right? Can we, just like when you use a deep learning algorithm to play a very complex game, like Go or chess, can you actually leverage the deep learning algorithm to shed light on a strategy or on an insight that complements what the humans have known about that science domain all this time? So we always want to use these algorithms for discovery as well as for prediction. Finally, I think you'll also hear this from the other speakers, but there are beautiful paradigms out there that leverage structure in the data, right? Images have spatial structure. Time series data have temporal structure, and there's continuity in the data that can be leveraged to build more robust algorithms. Talking about robustness, you will hear some wonderful words later, but one of our panelists who's to come later has written a beautiful paper on robustness of models, basically pertaining to stress testing AI algorithms in pathology in the clinical workflow. So there are a lot of artifacts in these data sets that could potentially compromise the quality of the algorithm, both in its development or its deployment. How does one assess this robustness meaningfully to do a good job so that at the end, patient care is not compromised? So finally, these are scientific aspects, but there's also a cultural aspect that is slowly but steadily coming down, which is, can we identify and overcome functional barriers to meaningful data inference through sharing in useful manners? And one aspect of this is this notion of distributed learning or federated learning, where one might not be expected to share their data per se, but inferences from their data through model parameter sharing can still be done. And Dr. Mahmood has some beautiful work in that space. So I think with this, I will quickly go out to the three speakers and bring them to you. Dr. Faisal Mahmood is going to be our speaker, talking about multiple aspects from his work, followed by Dr. Lee Cooper, and then we have a talk by Dr. Anand Madhubushi from Case Western. So without any further ado, I will quickly introduce Dr. Mahmood. I'm sorry. Check. Dr. Mahmood is an assistant professor of pathology at Harvard Medical School, Division of Computational Pathology at the Brigham Women's. He received his PhD from Okinawa and was a postdoc at Johns Hopkins. His research interests include pathology image analysis, morphological feature and biomarker discovery, and he's affiliated with DFCI at Harvard, the Broad Institute, and the Harvard Bioinformatics and Integrated Genomics Facility. So with that, I'll give it over to Dr. Mahmood. Thank you.